have more than 35 national seed association as members, as well as individual company members. And so we are represented in the European member states, but also beyond. So for example, the seed associations of Switzerland or UK, of course, and also Ukraine or Turkey are also members of our association. So we are representing the full seed sector, so specifically agricultural and vegetable crops. So the ornamentals are also organized in a different association, but that's maybe a bit of a background where I come from and which perspective I will be sharing today. What we did last year, so we did a survey within our membership, and I will present the main results of this survey. But first, let me start providing a bit of the context. So what you can see here is our milestones in plant breeding picture. So just to provide a bit of a context, we are talking about new breeding techniques, but of course new is always relative in view of let's say the time we are talking about. Plant breeding companies or plant breeders have developed new technologies, new breeding methods already since a while, specifically after the discovery of the laws of genetics by Gregory Mendel in the 1866. So after that, plant breeding really got a boost of new breeding techniques because it was much more a science-driven exercise than it was before the discovery of the laws of genetics. And what we are talking about today mainly, when we look at the new so-called new breeding techniques, it's what we here summarized as precision breeding. So very many different kind of technologies which can also be applied in different ways, and which are more effective and more precise than older breeding technologies. And specifically when we talk about genome editing, and this is also what I will be referring to during the next, let's say 15 minutes, is mutation breeding. So it's the old mutation breeding technologies which we had developed already in the 1930s of the past century, where breeders used radiation or chemicals to introduce mutations somewhere in the genome because it wasn't really controllable, compared to what we talk about today. So genome editing, which is much more precise and helps breeders to introduce mutations at a specific target. And interestingly, I had a look what society was talking about when we had this old mutation breeding in the last century. And what I found was a very nice a book actually, which is also available online. It's about a lady, Muriel Howarth, and she founded the Atomic Gardening Society in the 1960s. And you can see her on this picture. And she published this book, Atomic Gardening for the Layman. And it was really it was a society and a community of very enthusiastic people who tried to introduce new genetic diversity by radiation mutation breeding actually. And they were really, it was kind of a movement during that time. And it was a lady, and again, it's two ladies when we talk about genome editing and targeted mutation breeding. And here it's Emmanuelle Charpentier and Jennifer Doudna, who, as Dennis already mentioned, won the Nobel Prize last year. So it's maybe interesting to see how things are developing. What we did in Euroseeds, and that was actually also in the context of the commission study on NGTs, because we were consulted as a stakeholder, and we thought, well, we need to find a bit more, let's say, facts and figures when we talk about the interest of the seed sector in applying new breeding techniques. And we did a survey within our membership that was individual company members. And we got a reply from 62 plant breeding companies of all sizes to our survey. And as you can see, dominating are the small and medium-sized companies. Only 10% of those are large, big multinational companies. And on the left-hand side, you can see also the headquarter location of those companies, which is distributed mainly in the western part of Europe, also some overseas companies which are member of Euroseeds. But as you can also see from this picture here below, the seed sector is very international. So if you look at the headquarters only, you do not really gather, let's say, the activities of breeding companies as such, because 98% of those companies which were replying to our questionnaires are acting on an international level. So they have either their R&D facilities or their breeding and seed production and sales activities on an international basis. So the coverage of our survey is much broader than those countries which are indicated by the headquarter location on this map. We asked the companies if they are currently having research and development activities with new breeding techniques. And here we really focus on new breeding techniques in the sense of genome editing, which allows introduction of new mutations. So we are not talking about applications which would result in a classical transgenic GMO. So really focusing on targeted mutation breeding. And here you can see that already today, the majority of medium-sized and all of the large companies have actually R&D activities in new breeding techniques. For the smaller companies, it's close to 50% which are already active in using those technologies in their R&D. We also asked, would you invest in the development with new breeding techniques in the EU market if the varieties would not be regulated as GMO, but as conventional varieties? So, and here you can see, of course, the specific effect here is on the small-sized companies, which would definitely increase their activities. Close to 70% of this, also of the small-sized companies, would then invest definitely in those technologies, but still also 21% are undecided. And some companies were saying, well, no, we won't. And when we had a closer look to those companies were companies which have a specific focus for the organic market, and they do not expect that those technologies would be accepted for the organic market, even if they would not be regulated as GMOs. We also asked the companies to indicate the top 3 factors which they see as most significant in limiting the potential use of the new breeding methods. And here you can clearly see that specifically all the aspects related to regulation, so regulatory costs, but also the public acceptance under GM regulation, so specifically related to labeling, of course, like we have it under the GM regulation, and then specifically also legal certainty and future regulations and timelines. So the uncertainty in the past, so because we had this discussion on how are those technologies regulated since 2008 already, and but also how will it continue in the future. That's a decisive aspect which somehow limits the companies to be active. And also for the small and medium-sized companies, the aspect of having access to relevant intellectual property is a relevant one which they see as potentially a limiting factor to use the technologies. In which crops and crop groups are companies currently active in research and development with new breeding techniques? And here you can see it's all different kinds of crop groups. So from fruits and vegetables up to sugar beet, but also smaller specialty crops like chicory or forage grasses, etc. are covered. So it's a huge diversity, not only by companies, but also by crops. And I did an additional assessment of all the applications that have been sent to the USDA MI regulated database. You might know that in the US, they have a kind of pre-consultation process, which is called MI regulated. It was established It's currently revised under the new laws which are implemented in the US. But until August 2020, the consultations which are in the MI regulated database were assessed by crops, and here you can see also here the diversity of crops is quite huge. So a different country with different regulatory system, but a similar picture, I would say. In addition, we asked the companies what kind of goals and traits are you following with new breeding techniques in your R&D process. And here you can see it's also a diversity of characteristics from agronomic value, which is mainly yield, up to food and feed quality, but also biotic stress tolerance, so pest resistance, herbicide tolerance and industrial uses are of minor interest, but then also abiotic stress tolerance like drought tolerance or heat tolerance, for example. And we compared our data with a study which was conducted by the German Julius Kühn Institute in 2019. They did a similar assessment on publications, which are available online from scientific institutes mainly. And here you have a similar picture, although the focus is a bit shifted, but still you also here see that what we here in view of classical GMOs, so that herbicide tolerance is the major focus, it's not true for new breeding techniques. And also here I did an additional assessment on the USDA MI regulated database, and here you can see there are more applications summarized under the category other. It's mainly industrial applications, but also like shelf life and digestibility. And you have less applications in some of the other groups, but also here it's a huge diversity. And again, herbicide tolerance is not the major focus. We asked the companies also, when would you most likely bring products to the markets that we result from those new breeding techniques? And we did not specifically ask for Europe, but on a global level. And so here you can see we might expect specifically within the next 5 to 10 years some products on the market. We also asked if there was a delay regarding market access due to some regulatory reasons. And you can see that the current regulatory situation definitely has an effect. So at least a third of the companies delayed their intention to release products due to the regulatory situation. So the picture might have been a bit different when we would face a different regulatory situation. So there is already an impact of the current ECJ ruling and the GMO regulation in Europe, but also in other parts of the world. As I said, it's a global picture, so we don't have a harmonized situation on a global level yet. We asked, did the R&D activities change after the 25 July ECJ ruling on mutagenesis breeding? And here you can see that still a number of companies kept their R&D activities up to speed, but also a number of companies, and it's around 40% specifically also of the small and medium size, they stopped or decreased their activities. And specifically the big companies, which have, of course, R&D facilities also outside the EU, they moved either their product focus or their whole R&D activities out of the EU. So that's definitely also something which would give those companies a head start in case the regulatory situation would change, because they are still much more active also on a global level. Of course, we have the whole political discussion in Europe around the farm to fork and the biodiversity strategies, some very ambitious goals as summarized here. And definitely from our perspective and from the perspective of our companies, of course, plant breeding can contribute to those challenges. And we've summarized this here. So very many different let's say applications and of plant breeding and improvement of specific characteristics can help to achieve the goals which are laid down in this farm to fork strategy. And of course, we fear that European plant breeders might be in a competitive disadvantage compared to their counterparts in other parts of the world, and that's definitely true when you look at this map. So we see a number of countries like the South American countries, but also North America and in some parts of Asia. The regulations are in a way that they exclude those applications of new breeding techniques which result in products which are indistinguishable from conventional breeding. And in those countries, definitely the developments could go ahead, but also, let's say, the still existing patchwork which we have on a global level is not, let's say, helping to speed up, because of course a number of agricultural crops are traded on an international basis, and also seeds are traded. 20% of the seeds are traded on an international basis. And of course, if the regulatory harmonization is not advanced, it's always kind of a limitation to apply those technologies and to develop products. Yeah, and with this, I would like to finish my presentation, and I'm happy to answer your questions.
Okay, thank you very much, Petra. That was a very nice presentation, and it was. I think it's an important survey that you've done among the companies, and I think it clearly shows how important legal certainty is beyond anything. I think we do have two minutes for questions. So, are there any immediate questions in the Q&A? What do you say, Matina? Is there anything?
Yes, there is a question. It is about the survey. So about the survey covering 62 plant breeding companies, is this the composition of your members? 10% large, 37% medium, and 53% small enterprises? Or this was the sample for the survey?
In this case, it was the sample of the survey, but still, if you look at the full membership also of our national associations, I think it very well represents also the sector. So it was quite comparable to the representation of the different company sizes within the breeding and seed sector in Europe.
Okay, thank you, Petra. Dennis, if we have a little bit of time, one more question. So, Petra, why do you think plant breeders are not using NBTs for producing herbicide tolerance? Is it about a technical limitation of NBT? Do you expect this trend to change?
No, it's not a technical limitation, because you can, of course, create also herbicide tolerances. But what we see is that the application of those technologies, specifically when the regulatory, let's say, burden is not that high, is much broader. So it's much more interesting for specifically also small and medium-sized companies to apply those technologies to create different kinds of crop characteristics. The herbicide tolerance, or the domination of the trait, the herbicide tolerance in the classical GMO scheme is definitely also a result of the regulations, because you need to see if you end of the companies behind. Of course, the big companies are also the chemical companies, so they provide both herbicides as well as, of course, the crop seeds. So there is also let's say, a complementary approach, but still the regulation has an impact. So meaning, if you invest in research and development, you look at markets. So where can I sell my product, and what are the costs associated to develop a product? And if the costs are high, you develop products which are which can be sold on let's say, a huge market, and also for crops which are kind of cash crops, like the hybrid crops like maize, etc. So and which can be sold on a global level. If you have lower costs, you can of course invest also in smaller crops, in traits which are, let's say, maybe for specialty markets only. and also which in crops which are grown maybe only in specific regions of the world. So you don't need to be in crops which are like these cash crops and very broadly used on a global level. But it's not a limitation by technology, definitely not.
Okay, thank you. To keep on schedule, I think we move on to the next speaker now. And the next speaker is Jon Falk from Saaten-Union Biotec. Jon has a PhD in botany, and he has worked, among others, with barley breeding at the Carlsberg Research Center before joining Saaten-Union Biotec as the managing director currently. So his areas of expertise include plant breeding, biotechnology, and research and development management. Jon, please, the floor is yours.
Yes, thank you very much. I'll try to start my presentation. So I hope you can see it.
Yep, perfect.
So I just try to get to the presentation mode. Yes, thank you very much for the opportunity to join this webinar, which I think is a very important task to go for. And it's a pleasure to give you an insight on how especially small, medium-sized breeding companies in Germany are looking at this new breeding techniques. And I would like to give you an insight about our ideas. One thing which is very important if you think about plant breeding in Germany is also you have to understand which companies you have in that field. So I would like to give you an insight. I tell you something about what we do ourselves, and then I would like to introduce you to a project which is organized by the German Breeder Association, which is called Pilton, and which might be important also in communication to society and politicians and decision makers. So what you can see here, basically in Germany, most of the breeding companies are small and medium-sized breeding companies. And that is also one reason why you could see in the graph you saw from Petra that there's so many breeding companies from Germany answering this question because they were interested in it, and that is explaining it. And you see this is a huge number. So it's not dominated by international breeding companies. It's dominated by small and medium-sized breeding companies. And Saaten-Union Biotec is actually reflecting this because we have been founded already in 1984 as at that time Saaten-Union Resistenzlabor, because it was at that time that smaller and medium-sized breeding companies identified that they need biotechnology in order to compete with the larger ones. And they founded a centralized lab where we provide service to these breeding companies, which are even competitors on the market. So being at the pre-competition level, they started to cooperate to make biotechnology available. We were so successful that in the meantime, we have about 70 employees, and we have two subsidiaries, one in West Germany and the other one in East Germany, in Leopoldshöhe close to Bielefeld and in Garterslee. So the main goal, what we're doing is we support the development of new varieties by biotech servers, which is mainly production of double haploids in combination with marker analysis. But another thing is very important as well. We have to develop R&D project to make new technologies available. And we do this in collaboration with our breeders, but we do it also together with other breeders and also with university academia. And just to give you an insight, the question if smaller breeding companies are also interested in new breeding technologies, we already started a project, a publicly funded project in 2016. That was a collaboration with the University of Hamburg, where we decided we need this technology. And it's not about just using it. You have to know how to generate plant out of, in this case, a single cell, which is protoplast. And it doesn't work with all cultivars. We are interested to work with the lead material, not with a model plant. It should be breeding material where we apply it. That was the first thing. The second thing is we wanted to make it transient because it was clear that from regulation, if you make a classical transformation where you introduce a CRISPR-Cas mutation and make then a nil segregant, so by segregating out the genetic the transgene you have to put in, it will always be a GMO in Europe. So it must be a transient approach. We took a more difficult approach because we wanted to get this accepted in Europe. So it's a transient approach, which we used actually with potato and rapeseed, and we succeeded generating hundreds, in fact, 1,000 mutated plants each, in order to see how this method is working. But in the meantime, as you know, the new regulation changed the view on it, and the question is, what impact did it have on the project? We finished this and we have this platform, but we did not continue, at least for those breeders which are just operating in Europe. So answering basically that what you could see also in the survey from Petra, and I also gave a feedback to that and saying yes, because we are limited to these cultivars which are also for the market outside of Europe. But still, we believe that this is of importance. So we kept on a little bit of it, but we didn't go on for full pressure. But something else came up in the discussion among the breeders, the German breeders, was that we need some positive examples in order to convince people that this is a good idea, that we need this as a tool to complement all the other tools, like Petra told us, all the other tools which help us to fulfill all the requirements to get the changes we need in agriculture, get a more sustainable agriculture. And this is one tool which can help us to achieve it. And among the German Breeder Association, the breeders decided to set up a pilot project to show that this is important and that we have a good example. And this project is called Pilton. And the idea is to establish a multiple and durable fungal disease tolerance in wheat through the latest breeding method, which means basically CRISPR approaches. And there you can also see the link to a homepage, because what we also thought, it's very important that we show transparently what is going on in the project and have regular update on the progress of the project and everything you need for communication. So please feel welcome to join and have a look at the page. And here you can see that among the breeders, there was a huge majority. It's basically more than 50 breeding companies contributing to this project. So there's no public funding. So the breeders invested money to set up this project to really go all the way through, show that the ideas the trade we're interested in is successful. We would like to have field trials to show that it's working in the field, and we also would like to go all the way to the registration. So really pushing and make this public and see what is possible. And you can see the list is long, and it's including large international breeding companies as well as smaller ones. So what is basically the goal? What do we want to do with this project? We want to demonstrate and quantify the potential of significantly reducing the usage of plant protection products. We had a closer look what could be a goal where we can see the benefits from it, and that is the one we went for. So we have ecological benefits by reducing significantly the plant protection measures we need in the field. Of course, it's good for the farmer because it's reducing the cost for him, and it's also beneficial for society because we want to have a more sustainable agriculture in the future. And we can also make sure that we in the future can provide society with wheat and the quality we need. And we want to demonstrate how available genome editing techniques can be used to achieve this, and also in the light of intellectual property and those implications, because we want to show what is possible, but we also want to have a look at it, what is related to it. So what is the basic idea about this? You can read a bit more if you go at this homepage, but basically the idea is actually quite interesting because we didn't go to mutate a receptor where there's direct interaction for the pathogen. The idea was a little bit different. I try to illustrate this on the right side with this hand-drawn graph. usually there's an immune response by the plant, and there's always a negative regulator and regulating and controlling the response. And this negative regulator also preventing that this is the immune response is going too high and too long and never ending. So it's a transient response. But the goal was basically, and there's some good indication from literature, if you inactivate one of these negative regulators, this response would be even higher and longer. So that we should be able to increase the tolerance of this plant. We took wheat for a very simple reason, because it's hexaploid and it's more difficult. Even with classical mutagenesis, it would be very difficult to achieve it, because you have 6 copies you have to mutate. Usually you would get it for only one subgenome, and then you have to combine it by crossings. In this case, we want to see, can we get this in one go and see if we're successful for that. And since there is not a direct interaction with the pathogen, we hope that this is a broad tolerance, not only to one pathogen, to a whole set of pathogens. And there's a list of examples like Fusarium, head blight, or leaf rust, or Septoria leaf blotch. And since it's not so easy for the pathogen to develop a resistance and overcome this resistance, there's a very high likelihood that this is durable. So it's longer and it could be sustainable. So another aspect, as I mentioned, we also are interested in to see the accessibility of the CRISPR-Cas technology for the plant breeding companies, because as you saw, we have a whole mix of small, medium, and large breeding companies. And we want to make sure that this technique can be available for all of them, and not just for the large international breeding companies. So we want this to be a test for it. So we want to have a look at the license structure of the providers and the requirement to get access to it. And as I said, it's very important that we want to show that it's possible also for small and medium-sized plant breeding companies, because we're interested to keep the structure, because it has high benefits for society. The output of new varieties coming out every year is larger if you have a huge varieties of different breeding companies focusing on different areas, even ecological farming. And also we're interested to have insight into how is the impact on the plant breeders' rights. You most likely know, all of you, that plant breeders' rights means that all breeders can take even the varieties from competitors and make crossings, and about half of new varieties registered are coming from crossings with material from competitors. So this is like an open source system, and we want to make sure that the system is functioning even with new techniques and new systems coming into the market. So in summary, to get an overview of it, we would like to show, I hope I could show you that new breeding methods can supplement the plant breeders' toolbox, as Petra also mentioned. It's not the holy grail, but it can help to really get the goals we want to achieve, and we have to be faster. And this can help us to achieve it, and we want to show it holds the potential for reducing the response time of plant breeding to achieve this challenge, because we have to be faster. And think about it, to develop a new variety, we are already in the range of 10 to 12 years. It's already 6 years for all the test field trials, and we have to be faster. And the Pilton research project, which was initiated last year, is intended to show the potential and clarify the accessibility, because we want to make sure that everyone can get it, and initiate a broader and science-based discussion with society by have a very high transparency to it and invite everyone to discuss about it. And we think it's very important that plants that cannot be distinguished from conventional breeding varieties should not be regulated at GMO. So that's something we want to achieve getting into a science-based discussion with society and also all those responsible for that. And results from this project, you can get access to it by looking at the internet page, and there you will see some movies. So there has been press conferences, there are regularly updates by movies, and you can see what progress we make, and the first plants have been generated. So next year, hopefully they can be tested in the field, and everyone is invited to follow this project. That's basically it. I would say thank you very much. I hope I could give you some insight that this is important for plant breeders in Germany, and we are willing to help to promote this new technologies.
So thank you very much, Jon, for this nice presentation. I think it's an interesting project, this Pilten. I think it's really a good example of the potential or actual use of the technology. So we are fine with the time. We have at least 3 minutes until the next talk. So should we go ahead with 1 or 2 questions? Matina.
Yes, definitely. So, so Jon, what is your thought on possible negative effects of knocking out or down negative immune regulators on overall fitness of the crop? You might play with the balance and make the crop invest less energy in growth.
Yes,
yeah.
That's obviously the first question coming up. Is it good if you stop this and change the regulatory system? But all indications we have so far, there are some publications about that showing that there is so far no negative impact. Of course, it's an R&D project. We had a very close look with some expert team to see we have to get something really convincing, and there must be a very high chance of success. But it's still an R&D project. But we are convinced from this that this idea is good and would be very interesting, and it could also have implications for other crops as well.
Thank you. And one more question. So due to what factors were potato and canola chosen for site-directed mutagenesis? Is it possible to implement this technology to corn, for instance?
Well, the problem is, as I tried to explain it in the beginning, so people think we want just to be fast and save money. You have to be able to generate these plants, and it's difficult and doesn't work for all crops. And we are a cell culture lab. We have 30 years experience with double haploids, and it doesn't work for all of them. Very simple. You have to develop protocols for it. And it took us 3 years to get it done for canola and potato. And we're working, of course, also on other crops, but that was our expertise. And of course, you start where you have your expertise, and it's challenging, especially if you want to have it in elite material, not some model plants.
Yes.
May I? There was a comment earlier that's saying that Swedish consumer studies, so in Sweden, they show more focus on the result of breeding more than the used technique, and there is a great change of attitudes in the last years. That is in a Swedish context, but would you say, are you seeing something similar in Germany or not?
I think depends on who is the person you talk to. I think society is divided there. That is my impression, but I've seen this question. I don't have a clear answer. I was surprised talking about this project also when I mentioned that we are doing it. We are representing small, medium-sized breeding companies. We invest money into it and saying, oh, this is interesting. Also that we go for the transient expression way, not classical transformation. And I said, okay, that's good that you incorporate this problem we have with GMO. And I said, okay, we accept you have a problem with it. We go the difficult way, but we want to show you the potential. And I always had a positive feedback. But of course, I think it's basically also a question of education. But Petra can answer this question much better because she is much more in communication than I am.
Only we can keep this, we can put it on hold for the Q&A session later, to not to run late, because we are about to move on to the next speaker. There are some more interesting questions also in the Q&A, I see, but I think we keep them for the Q&A later. So let me then introduce the next speaker. It's Dr. Mariette Andersson from SolEdits, the company. She has a PhD in biochemistry, and she has many years of experience working both in the private sector and also the academic sector, working mainly with tailor-made compounds in potato. So she's the co-founder and CEO of SolEdits, and combines this with a researcher position at the Swedish University of Agricultural Sciences. The floor is yours, Mariette.
Thanks a lot. Let's see, did I manage to share, and can you hear me?
Yep, we can see it and hear you.
Good.
Perfect.
So let's see if I can have a presentation view. Yeah, thanks a lot for the introduction and for the invitation. I will try to make a, in one way, rather long story pretty short, where we have developed or actually came up with a research innovation at the Swedish University of Agricultural Sciences that now has led to a commercial breeding tool that we use for improving potato varieties. And as you heard, I'm sharing my working hours between SLU and this newly founded company named SolEdits. And we work with potato, and we do that because potato is a very important crop. Today, it's the third most important food crop after rice and wheat. But we also grow a lot of potato for starch production. And especially in Sweden and northern part of Europe, potato is the main crop for starch isolation. And potatoes are very nutritious. It has a high level of vitamins and minerals, and also a very well-balanced protein. But at the same time, it also contains anti-nutritional compounds like alkaloids, for example. And you have all most likely heard that it's quick carbs that yields a high glycemic index when consuming a potato. A good thing, another good thing with potato is that it has a extremely high output, very high yield when you grow potatoes in the field. But it also needs a very high input of, for example, pesticides, water, and nutrition. So to sum up this very good crop, it also has some drawbacks that we can work with plant breeding to solve, so we can make this great crop to be even more nutritious and even more sustainable in cultivation and production. And potato is tetraploid. It means that it has 4 genomes, and these genomes are heterozygous, which means that they are looking different, the 4 genomes. This makes the traditional crossbreeding a bit challenging, and the development time for potato breeding is quite long. And if we could instead then add one or a few traits to an already good genotype, that could save some time in the development and the breeding of new varieties. And we can do this by using CRISPR-Cas9 to then induce mutations and add genetic variation in good elite genotypes without adding any new DNA. And we use a DNA-free genome editing method on protoplast, as you heard Jon talk about earlier. And I will briefly go through how this actually works. Because we take small potato plants grown in tissue culture, and we use the leaves. And we cut up the leaves and add some enzymes so that we get rid of the plant the cell walls, and then end up with potato leaf cells. This is what you can see here in this green band. If we look at them under microscope, they are rounded in shape, very nice looking, I will say. We take these purified cells, we design where we would like this genetic variation to be inserted or induced, and we add then this CRISPR-Cas9, the gene scissor, to make the cut exactly at the site we would like a mutation to be induced. This mutation or this cut is then repaired by the cell itself, but it repairs it sometimes a bit different than it looked originally, and it's this differences in the new genetic context that is the mutation that has been induced. The key here is that we have managed to get the protoplasm isolation and the transfection where we add this gene scissor into the cell to be extremely efficient. So almost all cells we add this CRISPR-Cas9 to, it is integrated in. We leave it there for very few, short time, only a few minutes. And then we make sure that the gene scissor is discarded. We make sure that these cells start to divide. And when it has divided for a couple of months, we make sure that it can induce a new shoot. So this is what you can see here. It's shoot that has been developed from a lump of cells. And this process takes about 3 to 6 months, a little bit dependent on the variety we are working with. When we then are looking at each of these shoots, we can see that we have up to 90% of mutations induced in at least one of the gene copies. So the frequency of inducing mutations here is very high. It depends also a bit on the variety used. So, but I would say between 40 and 90%, with 90% is the highest score we have seen. So because we have this very high frequency, we can also produce knockouts of genes, more than one gene. So we can stack genes quite efficiently. And our record is 4 genes or gene targets at the same time in one round of transfection. But we have not tested more, so it might be possible, of course, to add even more genes, stacking more genes. Then it's just a matter of how many shoots you are analyzing before you find your plant of interest. So because of this really high specificity and efficiency in the method, this tool can now be used not only for research, it can also be used as an efficient breeding tool. And it's genotype independent. So all varieties we have tested, we have succeeded in inducing mutations so far. It's a very robust method. I would then like to give 3 examples of traits we have developed using this technique, and it's examples of traits that are both good for the environment and for the health. I would like to start with the project that are most advanced is the production of a amylopectin starch. And here we wanted to solve a problem that the starch industry has. And it's when they are extracting starch and sell it as a food product, as a thickener, for example. A native starch has in a product a low storage stability. And to circumvent this problem, they are chemically modified the starch so that it behaves very good in food products, and the quality can be kept for a long time. And this is not very sustainable production. It's actually a lot of chemicals used. It's 4,000 to 5,000 tons of chemicals used every year, only in Sweden. That's 50 trucks. And we should also remember that Sweden have less than 0.1% of the global market of chemically modified starches. So it's a lot of chemicals used in this production. And we know that one of two starch components is the major bottleneck, and that's amylose. Because amylose is the bad guy here, creating this low storage stability. So what we have done is that we have inserted mutations in the gene responsible for the amylose synthesis, and then made a starch that only contains amylopectin starch. And with that simple cut, we can save a lot of chemicals during production. So these are, as I said, advanced, and they are lines, candidate variety lines are grown in field, has for several years, and are actually more or less waiting for some kind of decision how to deregulate this type of plants. My second example is an amylose potato, and now we're actually targeting the bad guy for the storage stability, the amylose, because that starch component has benefits for your health. Because if you eat a potato with high levels of amylose, you get a slow carb instead of a fast carb, and therefore yields a low glycemic index when you eat such a product. It means that it will provide a very more evenly distributed release of sugars to your blood, and you will feel full longer. It's of benefit, for example, if you have diabetes, but also if you would like to control your weight or eat healthier. But it's actually degraded so slow that it can be classified as resistant starch. And a resistant starch has a prebiotic effect, which means that it stimulates the growth of good intestinal bacteria. And this bacteria is very important for your health. It actually helps you to decrease the risk of getting some serious diseases. Here we have done the same thing. We have made like a simple cut in this time in two different genes that is responsible for the branching of the amylopectin. And what we do get is an amylose starch with an amylopectin that looks more like an amylose with longer chains and no branches. And we have managed to prove in previous experiment that these high amylose starches do have a clear benefit for your a clear increase in resistant starch, and therefore clear benefit for your health. But the same starch we have also been able to show is also good for the environment, because this amylose starch has beneficial properties as a raw material if you would like to produce a renewable and biodegradable plastic. This plastic also has a very good gas barrier, which means that you can actually pack sensitive products in this bioplastics. A drawback, though, is that it's water sensitive, which needs to be solved either through processing or that you use this starch in a multi-layer packaging material. Below here, you can see previous studies we did with different levels of amylose content in the starch, and we could see that the properties of the plastics improved with an increase in amylose content. The third example is the development of a non-browning potato, and this has been done in a very close collaboration with researchers in Argentina. And here we targeted a polyphenol oxidase, And this oxidase is catalyzing phenolic compounds to become brown pigments that you most likely have seen in potatoes, but you have most likely also seen it in other crops and fruits like apple and mushrooms and so on. This gene is highly active in the presence of oxygen, so it kind of increases this browning if a tuber is wounded either by damage or wounding during harvest or post-harvest treatments. So either these browning potatoes are thrown away or needs to be treated post harvest treated to reduce the browning, because the browning results in a loss of nutritional quality, but it also affects the taste and textures. So this is nothing that the consumer would like to have. Same thing here, a cut in the polyphenol oxidase gene inducing mutations. And I would like to show you a movie, because these potatoes has been grown in Argentina, and our colleagues there harvested the potatoes, and they just made some crush of the potatoes and left it in the air in access to oxygen for a couple of hours. On the left side, you have the wild type. On the right side, you have the mutated potato, and it's very obvious that it is a trait that worked very well. Sorry. Oops, sorry. Now I need to get back again. How do that? Do you still see my presentation?
Yes, we do.
Okay, good. So the three traits, example of the traits I presented is made as a research group at the university. So I would like to acknowledge all that has been involved. And on the left side, it's our intermediate research group, all skilled people that has been involved in this, but also collaborators from SLU Uppsala and Argentina and Swedish starch producers and Italy. So to come back to the company then, as you could see, we have an efficient method developed. We have also commercial traits developed in the potatoes. And this, and together with a lot of questions from different companies and research groups that wanted to have this method available, we decided to make it available to everyone within the potato business and potato research area through this company, where we then can offer this DNA-free genome editing in a cost-effective and cost-effective and time-saving service. If you would like to, if a customer or academia would like to add the time and cost for setting it up themselves. And we can offer then to introduce genetic variation and mutations in a gene of interests and produce new genotypes in a variety of interest. And we can also help with finding solutions to desired traits. Then it will be a R&D, might be R&D project, and we will of course then also include suitable academia partners and institutes. So the company is owned by two researchers, SLU Holding and the Swedish starch producers, and we are located in Alnarp in the very southern part of Sweden, close to Malmö and Lund. And we are now operating in full from 1st of February 2021. So it is the short story. And the long story is that behind this talented SolEdits team, you can see on the right side, there is a lot of experience. Together, we have more than 90 years working with potato biotechnology, which has been the key to this very quick implementation and transmission to a breeding breeding method as has been done. So with that, I would like to say thank you.
Thank you very, very much for this nice presentation, Mariette. And I think it's quite nice to see that some research going actually into innovation. So you see the clear link between the two and a real application in society, let's say, of research. Let's say take one short question.
Yes, so a quick question for Mariette for now is, do you see any adverse effects deriving from tissue culture on the plants generated from the protoplasts?
Yeah, we do. We do have some clonal variation in up to 20-25% of the regenerated shoots. This will never like develop into a commercial plant. No. So we do that, of course, and that we need to take high for when we are developing or developing like candidates for variety testing. Yeah.
Okay, thank you. So there are a number of questions now in the Q&A, but we'll keep them for the session later. So next, I want to introduce Irene Sacristan Sanchez, who is the head of Unit Biotechnology at the Directorate General of Health and Food Safety at the European Commission. She's leading the work on the report on new genomic techniques that was requested by the Council in 2019, and which I think we're all looking forward to with great anticipation. Irene, the floor is yours.
Thank you. Thank you very much for the invitation as well to join you all and give a little state of play on the study, which is almost there. We're just a few days away. So indeed, as you all know, the Commission is working on a study on new genomic techniques. We understand by these techniques developed from the time that the directive was adopted. I thought the directive was starting to be an old piece of legislation, but after seeing Petra's milestones, I realized that it's actually just brand new. But anyway, of course, you all know as well that at the origin of the study is the ruling of the European Court of Justice on mutagenesis, which concluded that organisms obtained through mutagenesis techniques developed after the directive was adopted fall under the requirements of the directive. The ruling was on mutagenesis specifically, but the reasoning of the court can apply in very much the same way to other techniques. And the Council reacted to this with this request to the Commission, because on the one hand, they acknowledge that okay, the court had clarified a point of legal interpretation and that at least there was legal certainty on that. But at the same time, that there are a number of questions and practical implementation issues that the national authorities were confronted with, and not just the competent authorities, but also the research, plant breeding sectors and beyond. So we have been working since the request in November 2019 on this study, and what we are trying to do, and you will see it in a few days, is we really have tried to take a snapshot of the situation of new genomic techniques in Europe at this point in time, looking at it from very different perspectives. One is the legal status and the implementation of the legislation, which is at the basis of the request. But we have also looked at the current state of play in terms of scientific and technical development and market applications. We've looked at safety and risk assessment considerations, what has been already assessed by EFSA, by national authorities. We've done an overview of what research has been done, is being done in this area. We have looked at ethical considerations and also at what work is being done at the level of member states in terms of public dialogues and public debate, which, as some of you have mentioned earlier, is also an important element in this area. So we've worked on the study in-house. We've had technical contributions from EFSA doing an overview of risk assessments available in the area, and also from the Joint Research Center, looking more at the state of the science and of research and development. And we have also taken into account other studies that have been produced in this area, even though they were not generated specifically for us, for our study. So from the group of chief scientific advisors in the Commission, from the European network of GM laboratories. We also refer to the very, very recent opinion of the European Group on Ethics in Science. So there's a number of external contributions that are also taken into account. And then very importantly, we have based a lot of our work also on 2 targeted consultations on the basis of detailed questionnaires, one to the member states and the other to a variety of stakeholders, covering from the plant breeding sector, research, but also the organic area, the GM-free sector, environmental NGOs, etc. So we try to cover all the wide array of interested parties in this field. And the study will also give a bit a summary of the different positions, which, as you know, in this area vary in particular on a number of aspects, such as how to regulate, safety, etc. So we try also to give a very factual and transparent view of all the different positions so far in this debate. And then, of course, and this links to the presentations from the previous speakers, we've had in recent times very important policy developments at the level of the Commission in terms of the Green Deal and the Farm to Fork strategy, which set very ambitious objectives in terms of climate change, food sustainability, food resilience, and which recognize the potential contribution of biotechnology to this objective. So we have covered that angle as well, and reflected the fact that there is, and you have given examples in all your presentations. There are strong claims in the products that are being developed about their potential contribution to the Farm to Fork Green Deal, but generally Sustainable Development Goal objectives. So basically, this is what the study will cover in terms of different topics and areas. We are almost there. We aim to transmit it to the Council and then publish it on our website by the end of April, as requested. The study will not only cover all these different areas factually, but we will draw conclusions. So we will try to put it all together and try to conclude on a number of key points. But the study itself will not contain a policy course of action. The policy response will be based on the outcome of the study. It will not be in the study itself. So what will happen next? As soon as the study is available, we want to present it and discuss it widely. We already have some dates in Council working parties and a later date in May when the Council itself will discuss it. We will go to the Parliament as well. We are organizing also an event with the various stakeholders that contributed to the consultation. So basically, we really want that the study serves as a basis for further discussion. This is an area, and again, a couple of you have mentioned it already, where we really need an open and transparent and inclusive discussion on the role of biotechnology in the EU, and on the contributions that it can make to global challenges, but at the same time addressing openly the concerns that a number of stakeholders also express, and also the need to ensure safety, which is a clear underpinning, for example, of the European food legislation. So we do hope that the study serves as a good basis to continue discussing with stakeholders. I think there is an important role for the plant breeding section in this debate. On the one hand, in terms of using the potential of innovation to translate it into concrete applications that benefit society, that bring clear benefits. And the previous three speakers, you have all emphasized this and come with some of such examples. But I think it's also important that we all, and I think we all have a responsibility in the institutions, stakeholders, and so forth, to have a public debate that takes on the benefits, the concerns, and that helps us shape the next round of policy in this area. So this is what I can say at this stage. Unfortunately, we are a few days from releasing the study, and I am not able to go into the details of the conclusions and so forth. But I hope at least this gives you an update of where we are. Thank you.
Thank you very much, Irene. It was very interesting to hear, and of course we understand that it's very close now to being delivered, so you can't go into all the details of the conclusion, but it was very interesting and important anyway for us to hear about the current state of play. We have now about 15 minutes for a Q&A session, and I think there is quite a lot of questions in the Q&A, so shall we just dive straight into the questions, Matina?
Yes, so first of all, so we'll try to group them a little bit as themes. So we have a couple of questions.
Sorry, may I also say, I forgot to say before that all the attendees, they can upvote on the questions. So if there is any question that the attendees think is very important, they can put a vote on it to upvote, so to get it higher up on the list, let's say.
Yeah, but we should be able to take, to address most of them. So first of all, a couple of questions with regards to what the technology enables us to do. So are there any technical limits, and are there any properties that cannot be changed or introduced with CRISPR? Who would like to take that from our panel to start with? Mariette, Jon?
Ladies first, if you want to start.
Yeah. So I thought it was more or less two questions there, perhaps. But let's start with if it's something that we cannot do. I see this as a complement to other breeding technologies. It would definitely not be the one and only method for breeding crops in the future. This is one method, and a method that is needed. Because it can come to some traits, but it's not beneficial to use for perhaps other traits.
And Jon?
I totally agree. I think it's very important to say that this is just an additional tool which can make it faster, but you already need all the other ones. Good example, genomic selection, looking at haplotypes, the genotype background has always an impact on traits. If you have quantitative traits where you have up to 100 loci having impact on it, this genome editing will increase the variation, the haplotype number, but you still have to use all these techniques. I think that's very interesting also about plant breeding. It's very innovative. You need all the new tools, also big data, phenotyping, whatever. And since every plant is very, very expensive and valuable for the plant breeder, that's a difference to the farmer. A single plant doesn't cost anything, but for the plant breeder, one plant can cost a lot of money. It's worth spending money into innovation in all different kind of fields. So we can be the driver for it. And that makes it so exciting to work in this field. And as I mentioned before, it doesn't work for all crop plants. So genotype effects, also cell culture, it doesn't work. So there are always some limitations.
So following on from this question then, in terms of the limitations of the technology potentially, but there is a more general question here. Would you say that CRISPR has an absolutely necessary role in adapting to climate change? Or should we rather change our way of agriculture to adapt it to the changing climate? Or is either or maybe not? Petra.
Yeah, maybe I start maybe from a more general perspective, but if the others want to contribute also maybe from a more technical perspective. So I would say it's not either or, it's both. So breeding, I mean, breeding is improving the genetics, but of course you can grow the plant in many different ways. You can do it, I don't know, in conventional agriculture, you can do it in organic agriculture or in something in between. You can, so that's a plant is always performing in its environment. And that's also why putting plants in the field is so important during the breeding process. I mean, you can have the best genetics, but if it doesn't work in the field, it doesn't have a value. So we maybe a nice a picture which we created some time ago. We call it innovation in rubber boots, because you can have all these innovations, but in the end, it's the performance in the field. So the breeder in his or her rubber boots who needs to test the plants in the field, and that's very decisive. So it's from the breeding perspective, it's both. It's lab and field, but also then if you go to the agricultural environment, of course, improving agriculture and making agriculture as such more sustainable, improving soil health, etc. This will all contribute to, in the end, be more sustainable. It's not the one or the other.
Thank you, Petra. Mariette, you're nodding.
I just agree. Yeah.
Okay, so we have a couple of questions with regards to IP. So, for instance, there is here one direct for Mariette to start with. Do you consider IP rights as important when starting trait development projects in potato, for instance? And are these ensured for your current projects?
Yeah, of course it depends on the trait and also what to say. If we look at from the company point of view, if we are working with academia, if we're working with another breeding company, of course it is important with the IP traits, and that needs to be discussed together with the customer, the IP. Yeah, and we need to make sure that we have the right IP when we are producing varieties that might be commercialized.
So you have some perspective for varieties, to be of potatoes to be commercialized. Are there any in the market, and how do you set the price in the market so that it can compete well with conventional potato?
That's no on the market yet.
Thank you. So perhaps for Jon, Petra, are the German breeders concerned about the fact that mutagenesis can be IP protected through patents, and that this could potentially reduce the importance of UP or PBR? Could this have an impact on the breeders' exemption, what you call open source?
Well, I can start a few because I named it open source. Of course, this is part of the PlantEd project because we want, this is a topic we have to look at, and patents are already reality, and that's okay. The breeders are saying if this is limited to technical innovations, so basically it's connected to traits, and it works. It can work. There are some good examples in the past. But of course, we're interested that this kind of license fees you have to pay doesn't destroy the system. Because I think the progress we had with the breeders' rights and the breeders' exemption is such a successful story, and it's in the interest of society to keep this up running. And therefore, all the breeders, also those which have patents on that, agree on this, because this is a driver. As I told you before, new varieties coming to the market, half of them are made from crosses with breeding material from competitors. And that's driving the market and the success of it. And we have to keep an eye on it, but it will be possible. And the past has shown it.
Yeah, maybe I can just add on, because I think it's interesting to see you have heard Mariette, who is, of course, as let's say, a startup company developing traits, of course, also have an interest in IP protection. So it's not only an issue for the only big companies, it's an issue for, let's say, all breeding companies, the one way or the other, IP protection in view of plant variety protection or patents, but also it's a matter of getting access to relevant IP. So it's always finding the balance between protection and access, and the breeders' rights very carefully acknowledge this protection wish of the breeder in exchange to getting also access to improved material to further improve it. And the same needs to be found for IP protection by patents. So because patent protection is a bit more, let's say, restrictive regarding access compared to plant breeders' rights. But there are, as Jon mentioned, nice examples how this can work. So for example, licensing platforms or something like that. But this needs to be organized, and the interests of the different stakeholders need to be recognized. So it's a challenge also internally, so within the sector, which is discussed, and there are solutions which are, let's say, on the table and which are discussed, but definitely it needs further considerations. Yeah.
I can just add there or agree that what I meant previously was that we need access to IP and licenses. We will never be able as a small company to have IP rights on all the traits we will work with. So that's, I hope I got that clear now.
So one question here for Irene. Am I muted? No, for Irene. So has the EC assessed the impact of the current regulatory framework after ECJ ruling on mutagenesis on the EU public research programs and institutes?
We have actually, so this was a specific question in the questionnaires, both the questionnaire to member states and the questionnaire to stakeholders, to try to assess the impact on research. So we will report on the results of the information that we collected in the study. Yeah.
Okay.
Yep.
Thank you.
We are almost running out of time, but I think we can take one or two more questions. And I hope you all forgive us for running 1 or 2 minutes over time today. So we can take another question.
Yeah. So the questions are being upvoted while we are talking. So I'm looking here for the most popular question. So it's a live challenge. So let's see.
One popular question for Irene. Should we take that one? Another one for Irene. So when do you expect to have a law in place in the EU on new breeding techniques? Sorry, Matina.
No, that's the one. So when, Irene, when do you expect to have a law?
Well, for now, we have announced a study. We have not announced a law.
Yes.
So I cannot say more than that.
No.
We will have a study by the end of April, and any follow-up policy steps will be announced as a follow-up to the study.
Yeah. You also answered a second question, which was, what are the next steps and what would be the roadmap? Yeah, that would come with the announcement of the results of the study, I suppose. That's the bottom line.
Yeah.
Yeah.
Is there any more burning questions now? So we take the very last question.
Yeah. I think we have addressed more or less all the questions.
Yeah.
There's no other burning question. We addressed the technical, the policy, the IP, for the time that we have.
Okay, so yeah, we have run 1 minute past the schedule, so I will not be long now. I'll just round off this webinar a little bit. So first of all, I would like to thank you very much to all the speakers and the 4 speakers, plus also Matina for helping me today with the Q&A. So it has been really interesting presentations, and I really enjoyed listening to it. And I think the technology, gene editing, and also many different new breeding technologies, they're advancing very, very fast at the moment. I think there is a new paper every day about some technical improvement of CRISPR, for example. So I must admit, as a researcher, even I find it a bit hard to keep up with all the progress there is all the time. There's a lot of progress. So it is a challenge to keep, I would say, to keep the public informed about all the progress, what is happening with these techniques. And it's a challenge to keep legislation up to date with all the technical progress. But I would like to add at the end that this cost action PlantEd and also the initiative Växtnoden in Sweden, we are doing everything we can. We are doing our best to provide this kind of information about the progress in plant breeding. And this time for this webinar, we invited mostly the private sector, but for the next time, we will invite other stakeholders. So I want to thank you all attendees also for listening to this webinar, and I hope to see you soon again on another webinar, or perhaps even in a physical meeting eventually. So thank you all the speakers, and thank you all the attendees.